Hello, I'm Dr. Charles Knight, and this is another installment in my 10 Lectures on Evolution series. It's a continuation of what we did in winter quarter, now with the summer class, summer 2025, where I'm introducing some more papers and using some of the ones from winter quarter, but encouraging and helping my students to develop similar presentations that we've done in the last half a year or so. So this is another example on a new paper, and you remember I'm Dr. Charles Knight. I work at Cal Poly in the Bailey College of Science and Mathematics at Cal Poly in San Luis Obispo, California. I work in the Department of Biological Sciences, and again, these lectures are for my evolution class in the summer quarter of 2025. I want to remind the students that each of the slides in their presentation should have one of these icons associated with it, and these are some parts that the students could include. They don't have to include all of them, but it'd be nice if they included a lot of them and are presented in similar ways that I do in these presentations. So I'd like to start the story, this paper, with a little bit of backstory. I was introduced to mollusks in a kind of indirect way. I was interested in marine biology as a student, but I grew up in Wyoming and didn't have any access to the ocean except when I visited my grandparents in Florida. So that helped me continue thinking about the ocean as a great place of wonder. But then I ended up studying plant biology and took a class when I was in graduate school on phylogeny, and a speaker came and gave a presentation about phylogeny involving mollusks. And he had the most beautiful pictures of these organisms that were pretty foreign to me, and they were all mollusks. And I'm showing you this slide, which is full of different types of mollusks, and they're amazingly diverse and beautiful, and are worthy of study in their own right because of their morphological complexity, but also they're just very unique and special and amazing, I would say. So this slide is just supposed to get you interested in mollusks of great diversity, lots of different names for different groups, but a new paper came out that kind of clarifies the phylogenetic relationships of these species, and I thought you might be interested in it. So this is the paper title. It's a genome-based phylogeny for Mollusca is concordant with fossils and morphology. It was published in Science in 2025, and that was actually on February 28th of 2025. A little bit older than what I'm asking the students to do. The students have to pick a paper after April 1st, but I can go a little bit further back because I'm the instructor. So I want to start with my take-home points before we get bogged down in some details of this paper. So first of all, octopi, squid, and gastropods and bivalves are all closely related, but understanding the internal relationships of the 8 monophyletic mollusk groups has been difficult. Scientists have known for a long time that there's 8 groups, and they're all monophyletic, meaning they're all each other's most closely related ancestors. There's 8 groups. That's been known for a long time from DNA and fossil evidence and anatomy. But the relationships among those groups has been difficult to discern, probably because they differentiated very rapidly during radiation, maybe all the way back in the Cambrian, but we'll look at the dates here soon. So Mollusca genomes have a high degree of heterozygosity and many repeated sequences, and that makes molecular phylogeny and comparisons difficult because repeated sequences kind of lead to mismatches in DNA sequencing and also alignment programs, and can throw a monkey wrench into phylogenetic reconstruction programs. Here, analyses of hundreds of conserved BUSCO genes across hundreds, actually I think 97 or 87 genomes of Mollusca, reveal a new internal topology for Mollusca, and that's really the take-home of this paper. It's a lot of work to get there, and the conclusion is kind of like, see, we found it out, we figured it out. It's not an experimental study, but still very important in its own right. So what we can tell is that Mollusca experimented with a variety of body forms in the Cambrian, and understanding the relationships between these 8 groups helps us understand life's possibilities. I stole that phrase, understand life's possibilities, from the paper. I thought it was a good way of encapsulating the importance of this study. So I'm going to highlight some words here that I'm going to go over in my glossary notes slide. And heterozygosity, BUSCO genes, topology, and Cambrian are all important. But before we do that, let's talk about a few of the scientists that participated in this work. Julia Swigert and Su25 were the first and last author, respectively, in this on this paper. There's a bunch of other people too. They both work at the Senckenberg Biodiversity Museum. Probably has a different title than that. In Germany, it's in Frankfurt, I believe. That's Julia there in the yellowish sweater, and Su25 is the third from the front on the left-hand side. So this is part of their collection of mollusks, and Julia is world-renowned expert on mollusks, and now Su25 as well, I would say, because of this paper. She's a postdoc, or was a postdoc previously. I'm not sure what her position is right now at Senckenberg, but these are dedicated mollusk biologists. And this is an image of the Senckenberg Institute from a while back. But the German government has been so good at funding basic research for a long time. And this group has benefited from that, and other sources too. So as I do, I'm going to read the abstract slowly and carefully, since the authors put a lot of time and effort into coming up with or figuring out which words to include here, and science is so good at keeping it very condensed. I don't think there's a better way to start these kinds of presentations than just to read the abstract. So I'll read the title first again, and then read the abstract. A genome-based phylogeny for Mollusca is concordant with fossils and morphology. Extreme morphological disparity within Mollusca has long confounded efforts to reconstruct a stable backbone phylogeny for the phylum. Familiar molluscan groups, the gastropods, bivalves, and cephalopods, each represent a diverse radiation with myriad morphological, ecological, and behavioral adaptations. The phylum further encompasses many more unfamiliar experiments in animal body plan evolution. In this work, we reconstructed the phylogeny from living Mollusca on the basis of metazoan BUSCO, that is, benchmarking universal single-copy ortholog genes extracted from 13 new genomes, including multiple members of all 8 classes, with 2 high-quality genome assemblies for the monoplacophorans. Our analyses confirm a phylogeny proposed for morphology and showed widespread genomic variation. The flexibility of the molluscan genome likely explains both historic challenges within their genomes and their evolutionary success. Some more words that I think are important to point out are phylum, BUSCO, just the fact they confirm a phylogeny proposed for morphology and show widespread genomic variation, just emphasizing that point. And this may explain the challenges in reconstructing their group from molecular sequences, that is, the widespread genomic variation, but also perhaps their evolutionary success. Similar to the angiosperms that have a high degree of polyploidy and genomic variation, you can't argue, though, that angiosperms have been very successful, perhaps because of this genomic flexibility. So let me go over some notes. Heterozygosity is the average frequency of heterozygotes across loci, or to a different definition, the fraction of genes that are heterozygous in the genotype of the average individual. BUSCO is a benchmarking universal single copy ortholog. That's a big phrase. It's a single copy ortholog. Remember, orthologs are the same gene in different species. As soon as there's a speciation event, all of the genes are orthologs. Single copy orthologs are easier to study because there's only one copy. And then benchmarking universal are just indicating the conservation of this genes. And they are a tool used in bioinformatics to assess the completeness and quality of genome assemblies. So somebody might ask you, what's your BUSCO score after you've just finished sequencing a genome, which you may do in the not too distant future. But they can also be used for deep evolutionary comparisons and phylogeny, as is done here in this study. So BUSCO is a set of genes, and it's a tool, and it's used for deep evolutionary comparisons. I'll just say the acronym again: benchmarking universal single copy orthologs. Topology is the branching structure of a phylogenetic tree, and be careful, because topologies can look very different but actually be exactly the same. For example, all of these phylogenies are exactly the same. They have some different topologies in the way that the branches are, or the nodes are rotated, and the lines are drawn. Are they curved? Are they straight? Are they angular? And do the branches represent amount of evolutionary change, like in the lower right, or are they just positions, like in the lower left? Lots of variation, but this is all just variation in topology. Ultimately, these phylogenies all show the same relationships between species, so be careful. So, continuing with my glossary notes, the Cambrian is the geologic period that lasted from 538.8 million years ago to the beginning of the Ordovician period, 486 million years ago. It lasted for about 51 or 52 million years. And this is when multicellular life radiated to all of the major body plans, and that all happened in the Cambrian. This is the typical progression of geologic periods. The Precambrian is on the far left. The Cambrian, the C with a line through it, that's the typical symbol for the Cambrian. The O is Ordovician, the S is Silurian, the D is Devonian, the C is Carboniferous, the P is Permian, TR is Triassic, J Jurassic, K is for Cretaceous, starts with a C, but there's been so many other C words, and then CZ is the Cenozoic. So these geologic periods are defined by major mass extinctions. This was first discovered by Sapolsky and Raup in the 1960s. They plotted the total diversity or number of families of mollusks through time and saw dramatic changes when diversity increased, first right at the beginning of the Cambrian, and then decreased at the end of the Ordovician. That was the first major mass extinction. The dotted line indicates that. And then there's another mass extinction near the end of the Devonian, and the big mother of all extinctions at the end of the Permian, where 90% or so of families went extinct. The numbers there are a little, I've seen different estimates. There's an N-Triassic mass extinction, and then, of course, the KT mass extinction. We know it as the Cretaceous Tertiary, but it's been renamed these days to Cretaceous Paleogene. Maybe better just to say Cretaceous Cenozoic. But that's when the dinosaurs went extinct, or at least the non-avian dinosaurs, because the dinosaurs are still alive. They're flying around even today as birds. This graph is figure 1 from our textbook, but it's also a textbook tie-in, because basically all textbooks on evolution are going to have this Sapolsky curve in some way or another in that book. And I think it's a really fundamental thing to know as an evolutionary biologist what the Sapolsky curve is. It's a plot of changes in family abundance or diversity through time of mollusks. This particular curve has the ice cover extent in latitude shown as well in shaded blue lines in the background. I'm going to add now some text, which is just the Sapolsky curve It's a plot of Earth's biodiversity through time that highlights mass extinction events, primarily developed from marine fauna. This plot was the first time that biologists realized that there was something going on, some major mass extinction events, and basically the whole endeavor of life's history and biodiversity has been trying to figure out why there was increases in diversity, first of all, and second of all, why were there major mass extinctions. That's not what this paper is necessarily about, but we may touch on that in various ways this quarter. So this is the major figure from the paper. This is figure 1A through C. And as you note, figure 1A is what I was just lecturing about. And that was one part of this larger panel figure that has two other panels. And the main result of this paper is the figure 1C, which is the phylogeny. I want to point out a couple things here. This particular phylogeny is calibrated with fossils, which helps pinpoint the divergence times of some of the nodes. Those are noted by a black square with a fossil trilobite or some mollusk of some sort in there and a number, 525. You should be aware that that is in the Cambrian. That particular node is on the left-hand side of this phylogeny, and if you look closely, you'll be able to find it. Also, I'd like to point out that there are big dark circles, dark green circles, and those are the divergences of all the major groups within the Mollusca. So there's at least 7 that lead to 8 different groups. Those groups are shown on the right-hand side. And we're going to look at the names of those groups on the next slide, but they're color-coded, and they keep this color coding throughout the whole paper, which is awesome. Thanks for doing that. It makes it so much easier for my brain to track which group is which. Really, really appreciate that. There are also other time estimates from molecular clock estimates that are below that green circle. On the left hand side you see, I don't know if you can see my dot here. Yeah, I don't think you can, but the 485 is the molecular clock estimate for the divergence of that node. So there's 8 of those. Talking about a phylogeny, it's there, they did it. They have a number of taxa there, and all of those had whole genome sequences, but they didn't use the entire genome for this phylogeny reconstruction. Instead, they used the BUSCO genes, which were those single copy orthologs that were highly conserved and present in most, if not all, of the species. I wish that Julia and Zuen were here. I'd like to know more about the set of genes, like, for example, are the Hox genes in there? I have a feeling they would be. When I poked around to try to get the list of genes, it always wanted me to download the program, and I didn't really want to do that, and so I still haven't gotten the full list of the BUSCO genes, but I'll figure it out in some way or another, because I'd like to know, were the Hox genes there, or was it cytochrome C oxidase, or are they more housekeeping genes? To me, it's interesting. I would have appreciated a table of those genes, but I'm not a practitioner of the BUSCO genes, and I might be thinking about a little bit wrong. Maybe those BUSCO genes aren't really protein coding genes, but rather regulatory parts that are connected to genes. I doubt they're that conserved, but anyway, I'm kind of just riffing on some ideas there. So the main conclusion of this paper is that this is the correct topology of the 8 major Mollusca groups. The bivalves are related to the gastropods, but in the middle is this other group called the Scaphopoda. The cephalopods are sister to those 3 groups, and then the monoplacophora are sister to all those. Again, talking about a phylogeny is a little bit difficult because this is the correct branching order, and this is a major advance for the field of Mollusca taxonomy. And but for a group of students here in the summer that are not necessarily marine biologists, what do they need to know for the test? And certainly I don't think it's the names of all these groups. But I would like to point out that, like, this one, Solenogastries, is this tube worm type thing. It's pink, it's striped, it swims around, it's amazing. And the Polyplacophora are these guys, which are amazing too. All of these organisms are amazing in their own way, but they're incredibly beautiful, and they do things that we're just starting to discover. These are chitons, I believe. So I'm going to go back to my take-home points. Genome-based phylogeny for Mollusca is concordant with fossils and morphology. Octopi, squid, gastropods, and bivalves are all closely related, but understanding the internal relationships of the 8 monophyletic mollusca groups has been difficult. Mollusca genomes have a high degree of heterozygosity and many repeated sequences, making molecular comparisons difficult. Here, analyses of hundreds of conserved BUSCO genes across hundreds of genomes, actually I think it was 87 or so, reveal a new internal topology for Mollusca. And Mollusca experimented with a variety of body forms in the Cambrian, and understanding the relationships between these 8 groups help us understand life's possibilities.